Hi, everybody. Because we haven't had a whole lot of class time to cover this, I wanted to go over the basics of cell division on a whiteboard and make a video so you could see it. And before we get into the sort of vocab and all of that, it's important to realize that it's the big picture, like why you need to do each of these processes more than the words that matters. So if you can understand why we need to do each of these steps, it'll actually stick with you. All right, let's switch to the whiteboard. So we're going to be talking about cell division, and this is not a camera. This is supposed to be a cell with a cell membrane and a nuclear envelope. And within that nucleus, hopefully you remember, this is where you're going to have chromosomes. And chromosomes are remarkably long. On average in humans, about 100 million base pairs each. And if that's one chromosome, which it could be, there's another copy of it. Right? There are two versions of every chromosome. Hopefully you remember that. We are diploid organisms. So for every one of those, there's another one of those. That's two chromosomes. And here's a third and a fourth. Okay. And if I was going to be more accurate, I would have drawn 46 for a human, but we don't have any potential for doing that on this drawing. So we have a cell, chromosomes inside of a nucleus. And if we passed through the cell cycle, if we went from G0 into the cell division cycle, and we started to enter G1, it would look, something like this. Under a microscope, all of this stuff would be significantly more hazy, of course. Okay, if we entered into S phase, it would look exactly the same. You wouldn't be able to tell. Honestly, G1, S, and G2 are virtually indistinguishable visually. So under a microscope, you wouldn't see any of that. So that's why a cell in one of these stages, G1, G2, and S, is known as being in interphase. Again, it's an old term. It's an archaic term for the fact that, well, it doesn't look like much is going on. And so it incorporates G1, S, and G2 phases, virtually indistinguishable under a microscope. Stuff is obviously happening, though, right? Each one of these chromosomes during S phase gets a copy of it made. And so for each of these lines, there should be another one right next to it. And I should state here that each one of these lines actually represents a double helix and some proteins, because DNA is packaged in our cells. Chromosomes are about half DNA and half protein. But each one of these lines represents a double helix, and I want that to be clear before I get into the next steps. Okay, so as the cell cycle progresses, this cell may get larger, may not, depends, right? But maybe it gets larger, and I'm drawing this as sort of a polarized lengthwise cell. And there would be other organelles that would be growing in size and number and diversity as well as the cell goes through its cell cycle. The one thing I do want to draw in here, though, are what are known as microtubules. And maybe you've seen the videos where motors are walking up and down highways. These highways are called microtubules. And there's an area there where they're centered, and the microtubules might branch out from that location. You can imagine this kind of like a highway system. BART, for example, has a sort of a centralized hub. You can imagine everything going through city center, Oakland, which is true. Most of the train routes go right through there, and all of them branch out in different ways. So things can be moved towards or away from the central location. Okay. Now, what I want to do is show you what you've seen before, because I know you've seen this before. I know you've seen diagrams of cells going through mitosis, and it looks something like this, where the chromosomes are all lined up in the center of some cell that's often not even shown with a membrane. And then there are chromosomes lined up in the middle. And so we're going to have to figure out how to get to there and why we're even getting there. So there's going to be something like this. Okay. Something like this. Maybe something like this. And another one like that. All right. I should also note that oftentimes people will draw these incorrectly. They'll do something silly like this. That's just wrong. That's just wrong. There's no X. And we can get into that detail another time if you'd like. But sure, we're going to copy the chromosomes. They've been copied here. You can't see them especially well. That's happening during S phase. And when we go into mitosis, we're going to eventually want to line up the two copies. This is one copy, here's the other. And then we're going to want to pull them in opposite directions so that over on this side, you get one of these copies of that chromosome. And over here, you get the other one. And eventually, you'll build a nucleus here, you'll build a nucleus here, and then you'll split the cell in half. But how did we get to this point? How did we get to that point where everything was lined up? How do you move the chromosomes and all that? So let's take a step back. And I want you to see that all the parts are already here in this cell that maybe this is in G2 now. So let's imagine this is in G2 phase, about to enter mitosis. And you've got those microtubules, right? You've got those microtubules there ready to attach to chromosomes and then use motor proteins to pull them. You got the chromosomes there. And so it seems like we should be able to trivially just get into the next stage. Now, hopefully you've realized that there is something in the way, and that one of those things that's in the way is the nuclear envelope. So one of the things that we have to deal with at the start of this process is we are going to need to break down that nuclear envelope. So I'm just going to erase it here. It's actually ripped up, broken into, structures that are effectively large vesicles. They're not for transport functions. And these microtubules are going to have a role in actually ripping that nuclear envelope to shreds. But let's just erase it for now. Okay, so now we're all set. So now we can just grab those chromosomes and pull them. Maybe you've noticed other problems here. One is if you grabbed the chromosomes and you pulled them towards the center of that structure, well, they'd all go there. And then you could, you could cut the cell in half here, and all of the chromosomes would have ended up on the same side. That doesn't help us any. Okay, so let's say we were able to copy that thing. Let's say we had two of them. All right, so let's jump ahead and say, let's say, are there two of them? Great. So now we've got two of these. So now we're going to line up these chromosomes and then pull them after attaching them. So let's imagine they were attached. Imagine each of these chromosomes was attached. Well, hopefully you can just visually see that, like, if you yanked on these, they aren't going to go anywhere. And they might be tangled. You might pull on a little bit of the chromosome, but the rest of it might not go anywhere. It's just a giant mess of string or spaghetti. You might pull like one part of a string, the rest of it isn't going anywhere. So let's backtrack a little bit and start again. All right, we are starting again, and I'm going to walk you through 3 essential processes that have to happen at the beginning of M phase. So what we're going to move into is M phase, which is going to include mitosis, the splitting of a nucleus in two, as well as another process called cytokinesis, which is cutting the cell in half. So remember, we started with a nucleus, and within that nucleus were a bunch of chromosomes that were insanely long. So one chromosome, and it's been copied. So each of these lines is double helix. Another chromosome, its copy, which happens to be right next to it. Another chromosome, another one right next to it. Another chromosome, and its copy right next to it. So processes that we must do in early M phase. 1, break down the nuclear envelope. Nuclear envelope breakdown. Got to do it. If you want to grab hold of those chromosomes, you got to get rid of the membrane that's in the way. That's better. That's going to have to happen. Another thing that's going to have to happen, you're going to need another copy of this sort of microtubule organizing center. And so you're going to need a sort of a duplicate set of microtubules. I'm not actually going to list that here because in some cell types and some organisms that actually happens in an earlier stage. But let's just imagine that you are going to need another one of these microtubule arrays, these star-like structures known as asters. I'm not going to list it though. Another thing, those chromosomes way too big to pull. Maybe big is the wrong word, way too like extended and unwieldy. So what we're going to need to do is condense them. So we're going to do a process called chromosome condensation. Make them, I don't know, smaller, wrapped up, easier to move around. So if you actually condense these, using my fancy eraser here, that's what I've got at hand, we'll end up with, maybe that incredibly long chromosome looks like this, and here's its partner attached to it. Maybe another of those incredibly long chromosomes in here, and here's its partner next to it. Maybe one of the chromosomes was shorter. There it is. And here's another copy of it over here with its partner next to it. You'll notice I'm drawing one kind of solid, the other lighter, sort of empty inside, based on the idea that one of these is supposed to be maternal and the other is paternal. And if that doesn't make any sense to you, just remember that you have one copy of chromosome 17 from your biological mother, one copy from your father, biological father. And so that's what we're getting here. Okay, nuclear envelope, chromosome condensation. And then the third thing that we need to do is chromosome capture. So we got to capture those chromosomes on microtubules. Microtubules have a lot of motor proteins associated with them. There are a lot of motor proteins on these chromosomes themselves at a specific location that, various locations actually, that we don't have time to cover. So these are going to be captured. So all of these processes have to happen effectively within the same part of early mitosis. And so these processes of early mitosis have a name. And this is one of those vocab words that you'll get in textbooks or even in the standards list for AP Bio, and it's called prophase. But just use your logic, right? This isn't something fancy. This isn't, this isn't a magical concept here of prophase must be these things. It's just, if you want to get the chromosomes allotted appropriately, you're gonna have to get rid of a nucleus. You got to make the chromosomes well compact so you can move them, and you have to grab them. So that's the first set of steps. Okay, so that's prophase. That's at least the term for the beginning of mitosis. Now what happens afterwards is you're going to line them up. And that's going to be based on the action of a number of different processes involving motor proteins, which is well beyond the range of this class, but you're welcome to ask me about that another time. Right? And so these chromosomes are going to have to get lined up. And so they will, they'll be in the center. And the reason they're going to be lined up in the center is so that they basically can be apportioned to the two cells appropriately. They weren't in the cell, if they weren't in the center before you actually pulled on them, both of them might end up in the same cell in the end. Like when you split the cell in half, you don't want both of them in one area. So line them up at the center so when you pull on them, they end up separating into two different locations. So we're going to do two steps here. So metaphase was at the beginning. Those are those three steps of nuclear envelope breakdown, chromosome capture, and chromosome condensation. So sort of three processes that we talked about. The next stage is, oh, I just screwed that up. I should know better. I have a PhD in the study of cell division, and I already messed that up. This is prophase. That shows you how much I care about like vocab. All right, so prophase are those three processes. Metaphase is where you line them up. Line up, thanks to motor proteins and some other processes. The next stage, which is another vocab word theoretically to memorize, that's where you pull them apart. And so thanks to the actions of some proteins that cut other proteins, there are actually proteins holding these copies together. And these two identical copies of each chromosome are cut apart. The proteins binding them together are cut apart, physically cut apart. And then motor proteins and other processes pull the two copies apart from each other. That's basically the entire process, right? We can get into all sorts of details and we can give them fancy names, but the idea of just getting rid of the nucleus, grabbing the chromosomes, putting them in the center and lining and pulling them apart, at its base is hopefully an understandable topic. Now there are vocab words, prophase, metaphase, anaphase, and then there's one more that I'll talk about in a second called telophase, are these subsets of mitosis, right? And then there's cytokinesis where you actually cut the cell in half, right? And there are some other vocab words that are important to understand, and I'll give you one right here. These two identical copies of each chromosome are known as sister chromatids. But, you know, each of them is still a chromosome. It's just yet another vocab word to annoy us. Okay, I'll stop ranting about that. So what do we do at the end of this though? Like, we're not done just because we've pulled those chromosomes, right? So there is one more step. So eventually you end up pulling the chromosomes pretty far, right? You pull this one over here, this one over here, this one over here. Maybe all four of them end up in a pretty small area. And I'm not drawing 46. I don't care that much. So I'm not gonna draw 46. That level of accuracy is unnecessary. All right, so we're gonna pull them pretty close to that microtubule organizing center. Okay, so we're not done yet. We're actually not done yet. There is just a couple more things to note. Telophase is actually 3 processes, but nothing you have to memorize. Nothing you have to memorize because it's the exact opposite of prophase. It is the exact opposite of, you're just resetting the system, folks. So this is the opposite of prophase. Before you broke down the nuclear envelope, well, here you get to reform one. Before you attached the chromosomes to microtubules, well, now you get to detach them because you don't want them stuck to microtubules for the rest of the natural life of this cell, all right? And this is getting uglier and uglier, right? So let's detach the chromosomes. So they're no longer attached to this microtubule sort of star shape, this aster here, all right? So detach them. Make a nuclear envelope, reset the system, folks, reset the system. And then those chromosomes that have all been like shrink wrapped, well, unpack them so you can actually use them. It's actually really tough for a cell to survive this process in terms of like making stuff. When the chromosomes are all wrapped up in condensed form, you can't read them or use them from anything. So as soon as you've made your new nuclear envelope, you know, unpack them so you can read those instructions, generate the proteins you need to do your job, whatever your cellular job is. And then the last step, cytokinesis, which, you know, might not happen in certain cell types. You want to have certain cell types might actually have multiple nuclei. The last step is to actually cut the cell in half. And we're not going to go into the details of that. It should go without saying that anything physically demanding like this is going to require motor proteins. And so we'll end up with some sort of pinching action like that. So that right there summarizes the processes of mitosis and cytokinesis, as I mentioned below. And this is M phase. Hopefully that helped. If you have any questions, bring them to class next time. I will see you guys later.